should now go on with our next short presentation by Annamarie Dillon. She is coming from ProQR Therapeutics. It's a small biotechnology company in the Netherlands, and she will tell us something about gene therapy for Ashwaubenon type 2. So,
RNA.
You are ready? Okay.
Yeah, we're on. Everybody can hear me? Great. First, I'd like to thank the organizers for the opportunity to come here today to provide you a short update on our QR421A program that I'll walk you through in a moment. Just to let you know that as a publicly traded company, this presentation will include some forward-looking statements. So as was mentioned, ProQR is a Dutch biotech. We're based in Leiden, and our approach and therapeutic strategy within the company is to really try to have a patient-centric approach and focusing on rare genetic disorders where there is unmet need and currently no treatments available. It is also important that we understand the cause of the disease, for which we hope to develop RNA therapies that we will be able to deliver locally to the target organ. So in this case, it would be the eye. I included this slide just to provide a very high-level overview of drug development. It's a very busy slide, but it's just to highlight a lot of the different steps that have to be taken from taking a drug candidate through preclinical testing and all the different steps through clinical research and the studies that will have to be performed to better understand the drug candidate and to make sure that it is safe and that it works, and to be able to submit the dossiers to the regulatory authorities for their approval, and ultimately that physicians will be able to prescribe it for their patients. This is just an overview slide of our ophthalmology pipeline. So we have several programs currently in development at different stages. Our most advanced program, we heard yesterday for those that were there, is for Leber's congenital amaurosis type 10. With a compound called QR110, and it's exciting for us, but we'll expect our first clinical results of that later this year. And the next program moving towards the clinic is our program for RP exon 13 mutations. And we have some other work ongoing more in the discovery phase. So just a brief introduction to QR421A. It's a bit of a mouthful, I'll grant you that. So we're trying to develop an RNA therapy, so working on the RNA and not the DNA, to treat the eye symptoms, so the retinitis pigmentosa, for I'm not sure what ones are on there, but that will target mutations in exon 13 of the USH2A gene. So it's a targeted approach, and there's many different mutations within exon 13, and there is a publicly available database listing those out. And so building on the work, I think the sound is gone. It's back again. Building on the work from Erwin van Wijk and his team in Nijmegen, and we're moving this program forward. And this shows a cartoon. Here on the left is a healthy photoreceptor where there's no mutations, whereby RNA is translated into usherin protein, which is important for the maintenance of the photoreceptor. In the middle cartoon, I think here, yeah, we have a situation where there's a mutation present in exon 13, whereby the RNA is broken down, and this absence leads to absence of the usherin protein leads to retinal degeneration of the photoreceptors. Can people hear me? Because I don't hear something. Yeah, okay. And this third picture is just an illustration to try to show what our approach and treatment strategy is to remove exon 13 from the pre-RNA, so that the messenger RNA that will be produced will be slightly shortened, but we expect to be functional, so in a way to maintain the photoreceptors. So with all of the work that has been done to date, we are fast approaching moving this program into clinical trials. And the primary objectives of this clinical trial, which we're going to be calling Stellar, will be looking primarily at the safety, tolerability, and efficacy. We will be looking to see if it'll have some effect of QR421A in adults with RP and a mutation in exon 13. So I can only echo what Mariah mentioned earlier about the importance of understanding and knowing what your mutation is, because as more clinical trials, not only this clinical trial, but future clinical trials, we see more and more clinical trials focusing in on certain mutations. So we've, on top of doing all of the different safety tests and making sure that this is safe, we will also see if it'll have some impact, if that we can check that an efficacy with regards to vision. So I can't share a nice diagram of the clinical trial design today, unfortunately. We're still interacting with the regulatory authorities. So what we can say now at this moment in time, that we hope to finalize this very soon. It is good to mention that we envision that QR421A will be administered through an injection into the eye called an intravitreal injection. And we will also be looking at different dose levels within the patients. And for this first study, we will only include adult patients with mutations in exon 13. We hope, as mentioned earlier in the meeting as well, to move this program before the end of the year, that we hope to start it and start recruiting participants into the study, and we hope to be able to share more information next year, that we'll have some data, clinical data that we will be able to share. And the study, as foreseen now, will be conducted in North America and Europe. It's also very heartwarming as well to mention that we've been working and we have the support of the Foundation Fighting Blindness. They are supporting us financially with this program, and we are partnering with them moving this program forward. And also it's good to mention that we've been interacting with representatives of the Usher community. These past months, before we even thought about writing a protocol, we had an advisory board earlier this year with representatives both from the international Usher community, but also from several different countries in North America and across Europe, which was extremely helpful for us as well as we started thinking about the design of the study. And anytime we have questions, representatives that we reach out to are very gracious with their advice and support to us. So I wanted to say a big thank you for that. And if you'd like to stay updated on where we're at and how the program is moving forward, I would encourage you just to periodically check our website, as we will keep that updated with the latest information. And once the clinical trial is on clinicaltrials.gov, you'll be able to see there a full list of participating sites as well. So with that, I think I'm on time.
Much for this update. Yes, are there any questions?